Hi class, and welcome to this lab on fermentation, aerobic cellular respiration, and associated major organ systems. So for this lab, you'll be doing a couple experiments that I'll talk you through. This is laboratory 9. We'll do a brief introduction first of all. Aerobic cellular respiration functions to produce the molecule ATP, which is energy needed by all of your cells. It's needed to carry out all cellular work in the body, whether that's make proteins, um, create concentration gradients, and so on. The main fuel cells used in the process of aerobic cellular respiration is glucose. So here is the equation for cellular respiration. It uses glucose and oxygen to create carbon dioxide, water, and energy. So this is cellular respiration. Again, this is what happens in the presence of oxygen. They can use other proteins and fats, which eventually break down to glucose to go through cellular respiration. The reactions of aerobic cellular respiration, and the word aerobic just means that it takes place in the presence of oxygen. The reactions of aerobic cellular respiration always take place in the mitochondria. So that's the organelle in the cell, in the eukaryotic cell where aerobic cellular respiration takes place in the mitochondria. Oxygen is always required for aerobic cellular respiration because it accepts electrons that have gone through that electron transport chain. The breaking down of glucose in the presence of oxygen is the most efficient way to generate ATP molecules. And the most ATP molecules are produced this way, about 36 to 38. This is a great video that takes you through aerobic cellular respiration, and I'd encourage you to watch that because the last activity of the lab worksheet asks you to identify all the major organ systems and parts of organs and how they play a role in either cellular respiration or elimination of waste from the body. So take a look at that video. Cellular respiration and breathing are very intricately related, and that's because cellular respiration requires oxygen, which we breathe in from the external environment. Oxygen moves into lungs, diffuses across lung surfaces to the blood, and then is transported to cells. And then carbon dioxide is the waste that's created through aerobic cellular respiration that exits the cells, moves back into the blood, and then is carried back to the lungs where it is exhaled. So cellular respiration, the process of making ATP, requires oxygen that we breathe in, and then the carbon dioxide, the waste product of cellular respiration, will always be exhaled. Fermentation then. So what is fermentation? Harvesting energy from glucose in the absence of oxygen is fermentation. So fermentation takes energy from glucose, but without the presence of oxygen. Lactic acid fermentation occurs in some of our muscle cells and some bacteria. And the end products of fermentation always include carbon dioxide gas, lactic acid, and 2 ATP molecules. So it's much less efficient than aerobic cellular respiration, and that's because it's not using oxygen. So it's much less efficient. Alcohol fermentation then occurs in yeast, and the end products include carbon dioxide gas, ethyl alcohol, and 2 ATP molecules. So for activity 1, they'll take you through this process of fermentation in a yeast culture. Watch this video about fermentation. It's by the Amoeba Sisters. It's really great. Yeast are single-celled organisms in the kingdom of fungi, and the main fuel for fermentation is glucose. And as a reminder, the end products of fermentation in yeast will be carbon dioxide and ethyl alcohol, but in animals, it'll be carbon dioxide and lactic acid. So here's the fermentation with yeast. What we've done here is we've set up an experiment when we're collecting a gas at the top of this tube. And if you're wondering what gas that is, look up here to see what the end products of fermentation are. So that is the gas that's being collected at the top of this tube. On the next slide, you will see the results of fermentation using several different types of sugar and determine which to produce the greatest amount of gas or the end product, and that will signify the greatest amount of fermentation that was taking place. So here are the tubes set up, and the little yellow thing is the yeast. And then the gas is being measured in the tube at the top. And you can see where there's a greater white space, mean there's a greater distance between the yeast and the top of the tube. That means the greater gas. So your activity for your worksheet, and I'll just go ahead and show, let's look at the worksheet right now. So for the lab worksheet, you'll have to answer a couple introduction questions, which we talked a little bit about here. And then it'll ask which chamber produced the greatest amount of gas and which produced the least amount of gas, and what are the reasons that you can notate from that. And you can use, again, you'll have to use this slide to determine which produced the greatest amount of gas. All right, and if you have any questions about that, you should be able to get all the answers from either this PowerPoint or the worksheet. So I'm sorry for the extra noise that you might be hearing. They're doing some work on our house right now. All right, and then activity 2 of your worksheet has to do with aerobic cellular respiration in germinating bean seedlings. This experiment is using the uptake of oxygen gas by germinating bean seeds as an indicator that the process of aerobic cellular respiration is occurring, so in the presence of oxygen. We'll have a control with dry beans, meaning they're dormant, not growing, and germinating beans just means they are growing, so they will be uptaking oxygen for respiration. Dry beans are full of potential energy used to nourish a developing embryonic plant when it starts germinating or growing. While they are dry, they are dormant or not growing, so they would not be absorbing oxygen or releasing any carbon dioxide. So we'll always use a control with dry beans as a comparison of what should not be happening versus a germinating or growing bean plant. So what we've done is we will place the dry beans in water and some soil, and they will start to germinate or grow by using the stored energy in the process of aerobic cellular respiration. This produces ATP or energy needed to add new cells and produce pigments, and etc. While germinating, are they acting as autotrophs or heterotrophs? And I'll let you think of that. Are they producing their own energy, or are they requiring other things to create their energy? So this is the activity that we will kind of set up, and we show a picture of it. There is a link in the Canvas module that the link will take you to an experiment that shows this. You can go through that link, and I'll show you where that is right now if you're wondering what I'm talking about. So it'll take you to a link that looks like this, and then you can select the bean seeds and then do the same experimental setup. These are germinating bean seeds, and how the level of water will change. But what we've kind of done for you is we've drawn the experiment that we would have performed in lab if we were in person. And we've set up 2 kind of chambers here. The manometer tubing is filled with water as well to see the change in fluid that will occur. And here's the control with dry beans to account for changes in temperature and pressure in the room. And here are the experimental germinating beans that will actually be where cellular respiration is occurring. The paper wick with a vial of potassium oxide, that's KOH, this will absorb oxygen. So this will absorb carbon dioxide, that's a CO2. So this vial of KOH will absorb carbon dioxide, and then the change in fluid will occur here, and that will be for the experimental side. Okay. So how we've set this up, the control tube on the left side is with dry or dormant beans, and the experimental tube on the right side is set up with the germinating beans. Each chamber contains a vial of liquid KOH with a paper wick that has been dipped in the liquid and pulled partially out of the vial. And again, will be absorbing the carbon dioxide. And why is it absorbing the carbon dioxide? And that's because the experiment will use the uptake of oxygen by germinating seeds as an indicator that the process of aerobic cellular respiration is occurring. And remember, in aerobic cellular respiration, if we go way back up to the first slide, we see in aerobic cellular respiration, carbon dioxide is produced, water, and energy. So this carbon dioxide that will be produced from our germinating seed plants, the carbon dioxide will be absorbed by this paper wick in the vial of KOH. So that'll be absorbing the carbon dioxide. The clamp closes the system, and the manometer is a U-shaped tube with a red liquid that has scales on them to measure any movement of liquid that could be occurring. And any changes to the amount of gas in the chamber is measured by the movement of liquid in the manometer tubes. Why do we have a control tube? And this tube contains dormant beans. These beans shouldn't be absorbing oxygen or producing carbon dioxide because they're dormant, they're not growing. If there is a change in the gas levels in the control tube, this means that some other environmental factor in the room or something in the setup is causing a change. And we can assume whatever that environmental factor is, it's also affecting the experimental tube. So we must make an adjustment to any change recorded in the experimental chamber. And I'll talk about and explain that. So what we're doing here, and I'll talk you through kind of what activity will look like on your lab worksheet. You have to answer the questions on activity 2 in your lab worksheet. A few hints: we only want to measure oxygen consumption, not carbon dioxide production. And that's because that KOH is absorbing the carbon dioxide, but carbon dioxide must be in contact with the liquid to be absorbed. So what is the function of the wet paper wick? Well, the wick is continually to kind of take out that KOH and providing a liquid medium for the carbon dioxide to become in contact with. So that's why we use a wick in the liquid KOH vial. Germinating beans live off of stored food reserves until they produce leaves and can start photosynthesizing. So, and then in that case, with the germinating beans, we called autotrophs or heterotrophs. And again, I'll let you think of that. Again, an autotroph is something that creates its own food, and a heterotroph is something that lives off of other food. And the answer is right here. Germinating beans live off of stored food reserves. So that means they would be heterotrophs. But that only occurs until they start producing their own leaves and can start photosynthesizing. And at that point, they would be autotrophs. We are investigating, again, aerobic cellular respiration. So these germinating beans are using oxygen and glucose to create carbon dioxide and water. Do the germinating beans need to consume oxygen to do this? If it's aerobic cellular respiration, they do. And here's the hypothetical data on this page, which you will need to answer your questions on the worksheet. So these are different trials taking through the initial reading and end reading. And again, this is showing the experimental, the germinating beans kind of U-shaped tube and the difference in fluid level of liquid, because the difference in fluid level or liquid signifies the amount of oxygen that is being used. So let me just take you through this one more time. So let me take you up through these vials, these U-shaped tubes. You're reading a side of the fluid, and the fluid will go up or down. And if it goes, the change in fluid levels signifies that that's the amount of oxygen that's being used, because on the other shape of the tube, it's filled with oxygen. So as the liquid changes, that change in liquid volume that you can read on the side of the tube, we will correspond that with the amount of oxygen being consumed. And again, oxygen being consumed because we're doing cellular respiration and germinating seed beans. So they are consuming oxygen. So the liquid will change. And then they were creating carbon dioxide, and the carbon dioxide is being absorbed by the KOH in that vial. So anyway, what this experiment is showing, what this graph is showing, is you guys, and it basically does it all for you, but it's taking a difference of fluid levels and then it's multiplying by 7 millimeters cubed. What that gives us is that gives us the amount of oxygen in the final column. The control Readings should never change because the germinating or the beans in the control side of our experiment are not germinating, so they're not using any oxygen. However, if you do see a control volume change, you'll see the difference notated here, and then you'll see an adjustment done by the volume in the experimental group, because they're assuming that if something in the room made a change to the control, it'll also affect the volume in the experimental group. So pretty much what you guys need to be concerned with is just this final column. So these numbers right here, you'll be using the data from the table and the weight of the germinating beans, which we will give you, to calculate the number of ATP molecules produced per gram per minute by the beans. First, you just need to add up all the values on the last column of the data, and this is the total oxygen used in 30 minutes, and that will be space B on your worksheet. So you need to add up all of these numbers, and that's the total oxygen being used in 30 minutes, and put that in space B. For the gross, so then that'll ask for the net weight of the beans, and to find that, we give you the gross weight of the beans, and the tare, the word tare has to do with the fact that when we weighed these beans, they were on some sort of dish that also had a weight. So to find the net weight of the beans, you just need to take 37.38 and you have to subtract out the tare, the 1.7, and that will be space A. Place these values on your lab worksheet. Letters show where to place the values in the formula on the next page. And this is a kind of a representation of your lab worksheet. So the B is the total of numbers all on the last column. So letter B, you want to add all these up and get a total, and you'll put that in here. And then the net weight of the beans is taking 37.38 and subtract 1.7, and that'll be that. And then from here, it takes you through the math. And it takes you through the math eventually of how we're going to get ATP molecules produced by the beans. So put in B, put in A to get C. You have to do some division and multiplication, but I'll let you guys work through this. The directions are very clear. Here is where you get C, divide by 30 to get D. Put D right there, multiply 1.6, and that will give you E to get the total number of molecules per minute, and then answer the questions on your lab worksheet. So that takes you through pretty clearly how to do that. The last part of your lab worksheet then, I'm just going to move this out of the way because you'll have to identify the major organ systems involved in respiration process, be able to identify major organs on a diagram, and on your lab worksheet, discuss the basic functions of each structure, which this part you'll need to use your textbook for a little bit. So I give you a picture of the major organ systems, and let's take you through, I'm going to take us back to the lab worksheet. So the final activity, you'll need to use your textbook, and we'll also be going through this in lecture eventually. But all you need to do is explain what each organ has to do with cellular respiration, meaning how does your body get glucose? And in the digestive system, a lot of that has to do with eating. So what your mouth has to do with breaking down food into smaller pieces, what your esophagus does, what your stomach will do, your small intestine, the large intestine, a lot of the absorption of glucose into your blood occurs in these two places, and then where elimination occurs. So you're going to identify the major organs and how they're involved in the respiration process. And again, respiration means taking glucose and creating ATP. So the digestive system, its main job is to get glucose or food particles broken down into glucose. The cardiovascular system, its main job is to pump the oxygen that you are breathing in through your lungs and pump it out through the heart and blood vessels to take oxygen to all of your body cells that need the oxygen to do cellular respiration. Your respiratory system, what your trachea and your lungs do to get oxygen into the body, and what they do to get carbon dioxide out. And then the excretory system, this gets rid of all waste products. So whether that's carbon dioxide, which will be exhaled via your lungs, but also what your kidneys and your bladder do. So the waste products that your kidneys and bladder do. This was a little bit longer introduction, but I hope it will help you go through all the activities in this lab. And I hope you guys are all doing well. Thanks for listening.